the image reconstruction.
I will start. Hello everyone, I'm David from Tsinghua, and I'm actually really excited to be here presenting among the best students, so it's an honor. And I'm happy to be here with my collaborator who immensely worked with this project with me, Yuanzheng Ma.
Yeah.
And also I'm happy to have Professor Shun Guan. And also I want to thank Ma Xiaohua who supported with their advice and their work. So the title of this presentation is Exploring the Neural Organoid in High Definition. And in short, this work is about combining science and engineering. That's why it's really special to me. So let's start, right? Oops. Yeah. So what are organoids? Actually, organoids are a really great tool, and they are the best tool to use in biomedical research. But there's a thing, when we are studying organoids, there's a challenges in imaging and 3D modeling. That's why we are proposing strategy called layer link, and we are hoping that layer link will go beyond organoids and it will be used for cell-to-tissue organ and other 3D tissue bioprinting technique studies and applications. But I think we all need to understand what are organoids, right? So here you can see I chose this image of confocal image of cortical organoid, and this is really good because it shows the stem cells here and also mature neurons. So it shows that organoids have the capacity to show dual nature, both stem cell and maturity. What does this mean? It means that organoids can be used for developmental biology. Also, they can be used for disease modeling, and they also can be used to understand how different tissues are interacting with each other, okay? So, but as I said, the best way to study organoids is confocal imaging, but confocal imaging has its own set of problems. And one of them is that, yes, in a thin layer, we get the really nice images, but as the sharpness of images tend to fade as we are having the thicker images and the acquisition goes deeper and deeper. And itself gives another problem, that's that there's a crosstalk among layers. And this can be a problem or it can be utilized for our benefits, which I will explain later. And one of the things is, okay, we can do lots of layers for 3D space, but as we are imaging, we get the problem of photo-bleaching of fluorophores, which again reduces the signal-to-noise ratio. And here you can see simple experiment where we stained and imaged Alzheimer's brain for a long time of mice. And as you can see, this layer gets bleached. So, okay. So I talked a lot about everything, so now let's focus on our work, right? So as you can see in this schematic, first we generated organoids, which takes several months, depending how old you want your organoids. And then the most important one is that we are applying antibodies, secondary antibody, which is used for confocal microscopy. And this captures image that we are looking at. So we are looking at the information captured by secondary antibodies. And we did imaging for increments of 1 micrometer, and then we used layer link to increase the volume. So we decided to increase the amount of layers, and then we are presenting here 0.1 micrometer increments. And now I will dissect everything, but before that, there are key equations we need to consider. And the first one is weighted average. So we are trying to use weighted average for estimating and interpolating the layer we want to guess. And the best one so far we think is generalized normal distribution. So please keep these equations in mind. And let's discuss and dissect layer link. So first we are doing weight estimation for interpolation, and we are systematically modeling, and we are getting to get this generalized normal distribution. And this is a first initial estimation for structural similarity. And after that, we don't want to stop here because structural similarity is not enough. So we are using denoising neural network. In our case, we are using ResNet, which has own explanation. And we are iterating till we are getting the desired amount of layers. And one of the things in science we emphasize on is using ways to evaluate our method. And we emphasize on 2 ways. One is structural similarity, and another is peak signal to noise ratio. And we try to see how we can enhance this method. So we use generalized normal distribution for estimating neighboring layers, and then we use inverse function, which are comparative to each other, and then another ones we tested were exponential, cubic, and bilinear, which are traditional interpolation methods. However, we didn't stop here, and you will see in results that we decided not only use mathematical ways to enhance, how about we add further information to our model, right? So we decided to use neighboring layers, which are like twofold, fourfold, sixfold, and more. And we saw that beyond sixfold neighboring layers to get this weighting average, so we don't get like significant improvement. So here you can see in this F that we decided to take our layer link and extract the ground truth from it. And as you can see here, we don't have much random pixels, and this is very good. But here, in another case, we decided to use another diffusion model, and we extracted from that ground truth, and you can see lots of random pixels. It means that the gas layer was not accurate as we wanted. So, and here, as I said, we emphasize on PSNR and SSIM, and as you can see, first we try to do interpolation, and we get this structural similarity, and we want to make it enhance it, right? So after using ResNet, you can see the numbers are definitely increased, so this is something that makes us pretty happy. And finally, this one is something that really connects to me as a scientist. And this is about street reconstruction and seeing how it can be applied. So I'm studying gut-brain axis. So it means I'm looking at enteric nervous system and central nervous system. And here, as you can see, as we are adding more layers to this, as we are increasing the vertical resolution by guessing more layers, we saw that at some point we are seeing the neurons which are really important for us. As you can see, neurons are appearing, and this is really important because we want to see how, when we have systems biology, how each interaction is appearing, and losing the small signals is really, really big drawback for us. So this is one of the things that really excites me. And I want to make a conclusion and show up the hope and perspective. So one of them is that we believe we really speed up the data acquisition and improve axial resolution. And we are still continuing to do that. And then another one is uncovering hidden spatial information for systems biology. And for that, we are working on disease models to see how we can use our model to not only reconstruct, but also give a structure as a means of diagnosis. And finally, we are trying to see how it can be applied further. So I mean by